Good morning, good afternoon, and good evening, everyone, and welcome to this Inside Scientific webinar titled Multimodal Neuroimaging Using Functional Ultrasound, or fUS. I'm Andrew Davies from the events team here at scientist.com, and I'll be your host today. This webinar has been sponsored by Iconius, so a big thank you to them for helping make this event possible. We're very fortunate to be joined by Doctors Davide Boido and Antoine Bergel, who will discuss how state-of-the-art multimodal neuroimaging can be coupled with functional ultrasound to study neurovascular coupling across vigilance states. And with that, I'd like to welcome our first presenter, Davide Boido. Thank you so much for joining us today, Davide. The floor is yours whenever you're ready.
Hello everybody, thank you for watching this webinar on multimodal neuroimaging using functional ultrasound, ultrafast ultrasound. Let me disclose that this webinar is kindly sponsored by Iconius, but I have no financial relationship or interest with Iconius, nor I get any benefit or payment for this webinar. Every data I will be showing you today was recorded with a functional ultrasound machine that was a prototype developed by the group of Michael Tanter. In my presentation, I will address some questions on functional ultrasound with the help of multimodal recordings. In particular, how does fUS compare to standard neuroimaging techniques? And which is the relationship between functional ultrasound and neuronal activity? Next, what about the information carried by a single functional ultrasound voxel? Can we exploit it? Then you probably know about the high temporal resolution of functional ultrasound. But brain vascular activity is supposed to be slow. So is high temporal resolution really worth? And finally, what kind of signal is the functional ultrasound power Doppler? Is it just functional CBV? Let's go step by step, and let me briefly introduce the main feature of functional ultrasound. Without entering the complex mathematical details, classical ultrasound uses curved waves to get information on the sample, scanning column by column. Functional ultrasound instead uses planar waves to acquire the entire field of view of the sample at the same time, with a poor contrast with respect to classical ultrasound. However, functional ultrasound is so fast that we can afford to add information from many images obtained with different angles to recover a clear contrast and still preserving a high temporal resolution in the range of milliseconds. In addition, we can exploit the power Doppler effect, that is a modification in the US ultrasound wave backscattered by a moving particle to track the red blood cells flowing into the blood vessels of the brain. To get the answer of the previous questions, we developed a mouse preparation suitable for chronic recordings across many neuroimaging techniques: functional ultrasound, two-photon microscopy, and bold fMRI. We replaced the bone over the olfactory bulb with a special plastic cover that is transparent to ultrasound and light, and it is devoid of any artifact for bold fMRI. A step-by-step method paper for this preparation is indicated in the citations. Please note that what I'm going to show you today was collected on the same animal recorded serially, not simultaneously, across the different instruments. For simultaneous multimodal recordings, just wait the next part of the webinar by Antoine Bergel. I have used light sedation with methadone to avoid any confounds due to the different stress levels across neuroimaging modalities. So why the olfactory bulb? Because of the olfactory glomeruli all over the surface of the olfactory bulbs. These structures are made up by the afferents from the nose and the apical dendrites of the mitral cells. Glomeruli also have one of the highest density of capillaries of the brain, thus forming a functional neurovascular unit. Using mice genetically expressing a calcium-sensitive fluorescence reporter in mitral cells, the GCAM6, and playing with the odor molecules and concentration, we can see the activation of just a few or eventually a single glomerulus, as shown in the picture on the right. To be consistent across mice, we looked at the most sensitive glomerulus for a given odor, the thyltiglate, which is located close to the M72 glomerulus that in our mice was labeled in yellow. With GCAM6 and after injecting a red dye in the vasculature, with line scan 2-photon microscopy, we can record at the same time both the neuronal and vascular responses to natural stimulation. Upon another stimulation, the GCAM6 increases its fluorescence, while the red blood cells increase their speed in capillaries. Here the red blood cells are the black stripes because they do not uptake the red dye, and their speed is easily measurable from the slope of each stripe. We fix the odor duration and increase the odor intensity over a wide span of concentrations. And here on the right, you see the neuronal and vascular responses, both increase with odor concentration. Here instead, you see a coronal section of the olfactory bulb containing the most sensitive glomerulus for the tilting glade, with superimposed activation maps based on the percentage increase of the power Doppler signal. As expected, the higher was the odor concentration, the higher the power Doppler increase. We decided to average all the voxels of the slice and to track this value over time. Here below, you can see not the average of multiple odor representations, but two single responses for each tested concentration to point out the reliability of these measurements. Again, increasing the odor concentration, we get a bigger response in time and amplitude. Then we imaged in a second time the very same mouse under exactly the same experimental conditions with both fMRI, and we found a very similar global signal increasing with the odor intensity. Please note that the temporal resolution of both fMRI was lower than functional ultrasound, with 1 point every roughly 4.5 seconds. The most sensitive glomerulus for this odor was located somewhere on the first line of voxels, and we went imaging it with a 2-photon microscopy. We found that the microscopic vascular responses to odor had a strikingly similar dynamics to mesoscopic neuroimaging techniques, and of course, all the vascular responses followed the neuronal dynamics. Summarizing all the results from each technique, this plot contains the data from all the tested mice with 2-photon, and we found a linear relationship between the neuronal and microscopic vascular responses. Here you see the area of the responses of all the tested mice. The same linear relationship was found for power Doppler, and both fMRI was linearly correlated to neuronal activity as well. Altogether, these results show a fairly wide linear regime of vascular responses with respect to the neuronal activity. Probably now you are wondering about the origin of the axis, which is a point compatible with all the fits. The passage through the origin of the axis implies that any tiny neuronal activation should trigger a vascular response. I leave you with the curiosity. If you want to know the answer, please go look at the cited paper. Moving to the next question, what about a single functional ultrasound voxel? To tackle this question, we set up a proper co-registration of 2-photon and functional ultrasound, securing the fUS probe to the 2-photon objective and achieving a micrometer precision. In practice, we could make sure that the most sensitive glomerulus was exactly in the center of a given fUS voxel. Then we collected a complete database with 2-photon and functional ultrasound, finding a surprisingly high degree of similarity between these two techniques. In light blue, the neuronal activity, in blue, the microscopic vascular responses from a capillary, and in green, the single voxel or 6 voxels all around the most sensitive glomerulus, power Doppler signals. The high similarity was present independently of the chosen odor concentration and duration, from a single sniff stimulus up to 5 seconds, from a light odor to a strong one, and across multiple tested mice. So a single voxel power Doppler signal not only carries enough sensitivity to be studied alone, with no need of averaging on bigger ROIs, but it also follows extremely well the microscopic vascular dynamics of the most synaptically activated neuronal spot. Another advantage of having co-register data is the possibility to play the game, find the best voxel. In fact, since we know which is the voxel containing the highest neuronal activation, we wondered if a parameter exists so that we can point out this voxel from the power Doppler activation map. This game is a bit complicated, and let's make it easier, considering only the 6 voxels around the most sensitive glomerulus. And let's take also a simple parameter, like the area under the curve of the odor response. Here you see the false color representation of the power Doppler signal of the 6 voxels from 5 different mice. Now let's rank the 6 voxels based on this value. The first ranked voxel has the biggest response. If we could use this parameter to point out the voxel containing the most sensitive glomerulus, this voxel should always rank first. Indeed, here are the ranks of the 5 mice with many stimulation strands, and the rank seems to be randomly distributed. So the voxel doesn't seem to show a particular tendency to rank first. And again, this does not depend on the odor concentration or duration. It does not depend either on the chosen parameter, because we also tested, for example, a general linear model approach with similar results. In other words, because of the enlargement of the neuronal activation spot made by the vascular integration, we cannot map the activation as precisely as we wish. This also means that the functional ultrasound spatial resolution overtakes the maximal resolution allowed by the blood flow-based signals. Of course, this result is true in our experimental conditions and needs further confirmation to be generalized to other animals or brain regions. So, to answer the previous question, yes, we can definitely trust a single functional ultrasound voxel because it brings valuable information highly correlated to microvascular activity. However, because of the nature of blood flow-based contrast, we cannot get a localization power up to the most active neuronal ensemble. Moving to the next question, we know that vascular activation is supposed to be very slow if compared to neuronal activation, for instance. So is it worth to have a high temporal resolution as the one affordable with functional ultrasound? To address this question, I need to come back to 2-photon microscopy to evaluate the dynamics of the neurovascular coupling. We fixed an odor concentration and duration and collected a wide dataset from many mice, always from the most sensitive glomerulus for ethyl tiglate. Then we computed the transfer functions from each mouse data. And just to recall you, the transfer function is an example of model-free analysis. And it is a function of the time which, if convoluted with the input, in our case the neuronal activity, produces the output, the vascular response. The beauty of the transfer function is that it mathematically represents, in this case, the neurovascular coupling. Having computed a set of transfer functions, we looked for the best one. We found this one that I plot in orange, together with the canonical hemodynamic response function, the HRF, commonly used for human ball fMRI analysis. This microscopic HRF that we found is indeed very fast, with a delay of roughly 200 milliseconds and peaking at 1 second. Our best hemodynamic response function not only performs well on its own dataset, but it faithfully predicts the vascular responses on the other mice data, given the neuronal responses. And on the right, you have 3 examples. Now I have no time, unfortunately, to show you how surprisingly robust we demonstrated this microscopic HRS is. We tested it on different modulations of odor stimulus and also across brain states comprising wakefulness, always getting good predictions. Please refer to the citation for this information. We also made an open source software to compute your own transfer function on your data. And please let us know if you find something fast in your data too. Anyway, the fast neurovascular coupling makes it worth sampling the vascular activation with a high speed. But now the question is this fast neurovascular coupling detected also by functional ultrasound? So remember that we got co-registered microscopy and functional ultrasound data from the same mice. Here above, the neuronal responses, and below the correspondent power Doppler responses recorded in the 6 voxels ROI containing the most sensitive glomerulus. In red, the prediction made with the HRF that we found with the 2-photon data. In these 3 mice and in all the others that we tested, the prediction was very accurate. Moreover, I choose to show you responses elicited by a quite strong odor concentration. And you probably noticed that the predictions follow well the first phase of the responses, while the second phase is apparent, but it wasn't correctly predicted. Subtracting the predictions from the experimental data, we obtained these signals from all the tested mice, and we could hypothesize a non-neuronal or not local vascular activation with this late dynamic. So to resume, at least in our experimental conditions, the neurovascular coupling is very fast, and the speed allowed by functional ultrasound is not only adequate, but together with a well-calibrated transfer function, it might open the way to novel studies on neurovascular coupling. We reach now the last question of this webinar. What is functional ultrasound Doppler, power Doppler? Is it just functional cerebral blood volume? You probably know that because of its characteristics, power Doppler is not isotropic. In fact, it is only sensitive to the red blood cell speed component on the axis of the ultrasound probe. But despite this limitation, power Doppler signal could be exploited farther than just functional CBV. And here is an example. But first, let me recall you that the power Doppler spectrum is proportional to the speed of the scatter points. Higher frequencies report particle red blood cells moving at a higher speed. When we first started to analyze power Doppler signals, we used the ongoing approach consisting in discarding the low-frequency components of the spectrum. Any axial speed lower than 2.5 millimeters per second was trashed. Then we cross-correlated each voxel signal with a boxcar function representing the stimulus. However, this method didn't give us plausible results. In fact, these activation maps at increasing odor strengths are strange. The highest tested odor concentration seems to activate the olfactory bulbs less than a milder stimulus. So we used the microscopic vascular response from the same mouse collected with the 2-photon microscopy as a target function for cross-correlation. And we somehow fixed the activation maps. Now the highest odor concentration produces the strongest activation pattern. We also realized that our recordings were indeed very stable, with very little vibrations. And we could reanalyze the data, splitting the signal between low and high frequency power Doppler. The cross-correlation with the same target function gave us 2 sets of activation maps, one mainly related to capillaries and the other to bigger vessels with higher red blood cell speed. These 2 patterns, although sharing the same trend, do not perfectly overlap. This is to say that if we carefully filter the power Doppler signal, we can weight the functional CBV more on high or low blood flow vessels, paying attention anyway to the previously mentioned anisotropic feature of the power Doppler. To conclude, we saw that the neurons to vascular relationship at micro and mesoscopic level in the mouse olfactory bulb is linear. We can trust single voxel functional ultrasound because it faithfully reports the activation of the most active spot in the sample. However, the localization precision is limited by the vascular activation, which is wider than the neuronal activation. We also saw that the high temporal resolution of functional ultrasound is absolutely worth because of an unexpected fast neurovascular coupling, again, at least in our experimental conditions. And finally, we got a hint on how the power Doppler signal could be used to get more than just functional CBV. It's time now for the acknowledgements. Many neuroimaging techniques implies many people and labs joining their efforts. I thank in particular Sir Sharpak because most of the results I showed in this webinar come from my postdoc in his lab. Of course, this work couldn't be done without the help of my colleagues in the lab. I also thank Luisa Ciobanu at Neurospin for both fMRI experiments. And let me say that the choice of the olfactory bulb was very unfair for both fMRI. And finally, I thank all the collaborators whose help was crucial to set up the transfer function computation. And of course, thanks to Michael Tanter and his team for the help with the prototype of functional ultrasound machine. And of course, thank you very much for the attention, and I'm happy to answer your questions.
Thank you, Davide, for that wonderful presentation. It is now my pleasure to welcome Antoine Bergel. Thank you very much for joining us today, Antoine, and take it away whenever you're ready.
Yeah, so thank you, Andrew, for the introduction. I also want to thank Davide for his part of the presentation and introducing the important concepts of functional ultrasound. So Davide's talk was really about sequential multimodal imaging. I'm going to present my work on multimodal imaging in behaving animals, and we do functional ultrasound and electrophysiology at the same time. So this is a work that I did mostly during my PhD and then during my postdoc. So I was at the Tanter Lab and I had a great time there. We've done a lot of interesting stuff, and I hope that you guys will enjoy this presentation. So yeah, my talk is structured in 4 parts, and on all of these parts, I attached the different papers that I'm referring to. So here we go for the place of functional ultrasound in behavioral neuroscience. And when I started, so as a PhD student, and I arrived at the lab, I was at former Université Pierre and Marie Curie. I was with Dr. Ivan Cohen, who actually was collaborating with Michael Tanter. At the time, they did functional ultrasound in an anesthetized animal. I was super interested in the rhythms of the brain, the question of neural synchronization. So we were discussing this, and at the time, my understanding was that when brain neurons fire together, they create brain waves, and these brain waves, they are called brain rhythms. So basically, when you have correlated spiking in the neuronal activity, you have fluctuation of the electrical potential that is detectable by macro electrodes at the surface of the skull. And in turn, those oscillations in the field potential, they shape and they influence the activity of millions of neurons. So what was super interesting for me was the fact that using electrophysiology, you can detect and study those waves. And another very important stuff is that those waves, they actually offer a window to study cognition. So one of the simplest experiments that you can do with electrophysiology is to put a single bipolar electrode. Here it's in the hippocampus of rats. So this one was done in the early 70s. And you let the animal do whatever he wants to do. So basically at some point he will move, he will start walking, running. Sometimes you see it jumping. You can also place it close to water. You let it swim, climb. At some point, the animal will make pauses, right? So, and you confront basically the pattern that you see in the electrophysiology traces when the animal is actually engaging in some behavior and when the animal is still. And what you see basically is that you see a very rhythmic oscillation that's now referred to as a theta activity. So it's an oscillation around 7 to 8 hertz. And you see this theta activity every time the animal engages in exploration and during REM sleep. So we are going to see that in the second part of the presentation. However, when the animal is immobile or doing slow wave sleep, we refer to that state as an offline state, and basically you just see large irregular activity. So these two states, they're mutually exclusive. You cannot observe them together. So at the beginning of my PhD, the idea was, okay, we would like to study these brain rhythms. They are interesting to inform us on cognition, but if you only use electrophysiology, then you have a lack of spatial resolution. So not only you have a lack of spatial resolution, but also these brain rhythms, they're not local. If you put an electrode elsewhere in the brain, you will sense them and you will see them. And it was demonstrated later that especially theta activity, they're traveling waves, you know? So you don't exactly know where it starts, and it's not exactly the same pattern that you see in different brain regions. So we need a technique that can monitor theta activity and its propagation throughout the brain. And that's often what we use to introduce functional ultrasound imaging. But if you classify all of the imaging techniques that are available now, you see that basically you have two worlds. You have the world of global imaging that's able to monitor the whole brain, but most of the time you will lack either temporal or spatial resolution. And on the other side of the spectrum, you would have techniques like electrophysiology that are able to monitor the activity of neurons, but they are not able to have a large field of view. And what's super interesting with functional ultrasound is that it's basically sitting in between these two worlds. So our approach was, okay, let's try to combine electrophysiology to get those brain rhythms, to have the nice temporal resolution that it offers, and to have the large field of view and the versatility of functional ultrasound. And if you do this, basically when you get the two techniques working together, it's a super powerful approach. So you'll be able to see blood volume on a full slice of the brain. The spatial resolution will be 100 to 150 microns. And with the LFP, the electrophysiology, you will be able to have a super nice temporal resolution. So now comes the hard part. So to do that, you're encountering several challenges that I'm going to explain here. So again, there's a bunch of publications that you can refer to. If you have further questions, feel also free to send me an email or contact me if you need assistance or have specific questions on this. So challenge one, of course, is designing long-lasting implants. We do the kind of behavioral studies where we want to record and monitor brain activity across days, possibly weeks, and why not months. And most of the time, we have a learning phase. So the animal learn a task. Most of the time, we do it before the surgery. Then there is a recovery period, 1 week recovery period. And then you want to start your recording sessions. And depending on the difficulty of the task, these recording sessions can span for a very long time. So we had to design a surgery that was able to do that. And we have decided to do a cranial window, so to maximize the field of view that we can have and to replace the bone by a skull prosthesis. So that's what it looks like. It's about 4 to 6 hour surgical procedure. And you see a very clear window. You can see the brain. We leave the dura intact. And here you also see the wire and the electrodes that are plunging into the brain and the connector on the back of the head. So these electrodes, they have to be super thin to minimize the tissue damage, and you don't want them to interfere or to be on the path of the ultrasound waves. So you have to design them with an elbow, so that's handmade electrodes. If you were to use normal and standard electrodes, you would have to implant them on the sides so that at least the connector is not on the way of the ultrasonic waves. The second biggest challenge that you encounter when you do this, of course, is to not impede the free movement and the unrestrained movement of the animal. So throughout the years, there's been a constant evolution of bulky. We really started with bulky probes that was super hard to get the animal to move. And also, we could tell that the animal was not feeling well. So in 2012, that's what the ultrasound probe looked like. 2015, there was a first big revolution. We were using this second generation probes. Then 2017 came and we were able to change the holder. And finally, 2019, we really have this miniaturized probe that allow some applications to be done on awake mice. Also, you want all those images to be stable. So you want the probe holder to be completely fixed onto the skull and to the probe. So we use most of the time screws. You can also use magnets, depending on the application that you want to do. In this application on the left, we also had a motor that was allowing the probe to move to the single planes, to the different planes without requiring any intervention from the experimenter. And the last one, of course, is the cable. So the cables, they can be pretty stiff, but if you manage to help and follow the animal with the hands, there's also other systems that have been developed after that, but you're able to have your animal to roam around. It looks super free, can turn around easily, gets its rewards, and run and forage normally. An important point here is that we do not use the 3D probes that were used in other publications. They're not available on freely moving animals yet, and we end up with full planar sections of the rodent brain. So if you were to scan the whole brain, you would have to do it plane by plane in our setup. And the last challenge is that basically we have multimodal data. So we have electrophysiology, video, and ultrasound imaging, and all of these data is acquired by different computers. You want to trick them so that you can do the acquisition all at once, and you want after that to be able to analyze this multimodal data and to look at it, which is not easy. And most of the time, these tools do not exist yet, so we have to develop them, and that's what we currently do at the lab. So once you've overcome all those challenges, which looks very easy and I make it sound like it's only 5 to 10 minutes in this presentation, but it was actually 4 years of work. Now we can go to the applications, and I have two main examples to show how powerful it can be to combine functional ultrasound and electrophysiology. The first one will be during sleep, and the second one will be during locomotion. So during sleep, basically what we did was to place the animal in a box, and again, you let it roam around, you let it do its life, whatever it wants to do. And what will usually happen if you leave it like this for 40 minutes to an hour is in the first part of the recording, the animal would be active and awake. And at some point, it will stop roaming around. It will sit and start sleeping. So to monitor this, we have a bunch of sensors. We can monitor EMG, electromyogram, so that's the muscular activity in the neck. We have a miniature accelerometer. We have the LFP that I already introduced. And using standard sleep scoring methods, we are able to discriminate between 4 states. So these are the different colors. Green is quiet wake, blue is active wake, yellow is non-REM sleep, and red is REM sleep. And now if you look at functional ultrasound and the cerebral blood volume during these 4 different states, you do have 4 different pictures. Quiet wake is really quiescent. So you have large irregular activity on the electrodes, and you don't see much on the functional ultrasound images. When you have active wake, you have some activation, especially in the cortex. And you can see here on the left, the theta activity that I was talking about in the beginning of the webinar. Now, when the animal goes to non-REM sleep, it's still pretty quiescent. You don't see much, but in REM sleep, however, you see a huge increase of vascular activity almost everywhere in the brain. And at first, we were very surprised of seeing that. We thought that there was an artifact because it was really exceeding even what we saw during active wake, which is supposed to be a super active state. And so we put together, and we were really looking at these movies of the vascular activity. And that's what it looked like. So here I'm going to display a video. On the left, you will see the functional ultrasound images. On the left as well, you will see the video and the progression of the recording. And on the right, you see the electrophysiology, 8 electrodes here, the EMG, that are the gray electrodes, the accelerometer in the green, and the last one is the whole brain, the activity of the whole brain, and this is actually a measure that comes from the functional ultrasound. It's the mean of the activity in the whole brain. And what you see when you do this is that basically the animal is awake at the beginning, now it stops, and then you will slowly fall into non-REM sleep. And then at some point, when it's done a bit of non-REM sleep, here I decided to choose only this transition from non-REM to REM, we will see that the activity, the vascular activity goes super intense, and you will see the EEG, the LFP synchronize on the right, and you will see the emergence of theta waves. So here we enter REM sleep, and here we see that an elevation of the vascular activity that's super intense. And now you will see that it's not constant. There's like pushes and these moments where the vascular activity strongly pushes, like here. And again, here. So when you look closely, actually I'm going to stop the video and just go back a few seconds before. So before one of these pushes, you can see that there is something in the electrophysiological activity. Just pay attention here. So there's a pattern that we see. And we want to see and we want to analyze this pattern. So when we do that, we can do time frequency analysis, which is a standard technique in electrophysiology. This is a full REM episode, and we see on the bottom, we have the vascular activity, the CBV change, and we see these pushes, like these surges of vascular activity. And most of these surges, they are preceded by a specific pattern of electrical activity. And this specific pattern of electrical activity is stereotype. It's always the same. So it's like a bunch of, suddenly you have an increase in both the theta and gamma activity. So these theta bursts, they've been described during REM sleep. And if you look closely, intermingled in the theta waves, you have this very strong, peculiar oscillation that are super fast. They're about 100 hertz, and they are a very good proxy of neuronal activity here. And they always precede these strong hyperemic events that we call vascular surges. So if you want to analyze the correlations between the theta and gamma rhythms that you see before these vascular events, basically what you can do is to extract the peak from the electrophysiology in the different bands of your LFP and compare them to the peaks of the vascular activity. And if you do that, you find super nice correlations in the high gamma and the mid gamma band, but not in the low gamma band, and only moderately in the theta band. So that's really an argument to say that these extreme vascular dynamics that we see, they're really caused by electrophysiological patterns. The second thing that was also interesting to us is that if you compare the functional connectivity between the different states, you see that REM sleep, basically it's a whole red matrix saying that all of the regions, they're super synchronous. They all behave the same, except for one region, and you'll see that in the rest of the presentation. Actually, it's now. So here it's another project that was recently published last year. At the time she was a PhD student, now she's a postdoc at the Tanter Lab, Marta Mattei. And we had the strategy of, instead of staying and leaving the probe in only one plane to record brain activity, we really wanted to scan the whole brain. So it's difficult experiments because it takes some time. And Marta did an amazing job into scanning the whole brain, both coronally and sagittally. And what we saw is that we could reconstruct the full brain activity during REM sleep. And you see exactly the same pattern. You see that most of the time, the activity is moderate at the periphery, but it's super strong at the center part and in the other cortex region of the brain, here also in the thalamus and the midbrain. And you have a super clear view here, and you see the how powerful fUS can be, because it gives you information and you're able to scan like a very large number of regions. So we were super happy about this result. And another very interesting result is that when you were looking at all of these images and section, you were basically saying like the activation everywhere in the brain except in the posterior amygdala. So strangely, if you look at these correlation maps, you see that the posterior amygdala seems somehow disconnected from the rest of the brain. It's really this only region that does not behave like the other ones. So there's many interpretations to that. We don't want to rush into conclusions here. I refer to the paper. And again, there was a super nice work and super interesting conclusion in that paper. So the last example that I'm going to tackle here is this multimodal imaging during locomotion. So again, two papers that I'm referring to. So hemodynamics during locomotion, at the time we had another constraint, is that we could not image continuously. We had to basically decide when to start the image, the ultrasound scanner, and we had only 12 seconds. Now we can do it continuously. We don't have that problem, but that's what we had at the time we were acquiring this data. And so basically, I was trying to decide when the animal would turn and do a run, because you want to look at what's happening during theta activity. And again, that's happening when the animal is running. So we had this task when the animal was taking water rewards from one side to the next of a linear track. And here I will show basically the trajectory of the animal, the vascular activity in the middle, and the electrophysiology on the right. So here, that's one run, and you'll see the succession of the different runs. You see that we have clear signals. We do have some artifacts that we can reject. Most of the time, it's pretty clear. And again, I don't know if you can look closely, you can see the start of a sequence. So different regions that are activated in a temporal given order. And this sequence is actually starting in the central part of the brain and spreading to the hippocampus, and here we saw it pretty clear. So to analyze this sequence, another way to display it is just exactly the same, but instead of, we just like slow things down a bit. And here we'll have the trajectory of the animal on top. We'll have also the speed of the animal, and we can reconstruct the sequence of activations. And here you see very clearly that 1 to 2 seconds, and it peaks at 3 seconds after the animal started the run, that you see a very strong pattern of activity in the hippocampus. Another thing that you can do is to detect all of the starts and all of the ends of each run, and you can realign them on all rats, all recordings. And when you do that, you have basically, you can reconstruct the hemodynamic responses associated with locomotion. And at the time when we started this, some people were telling us, you know what, if the animal's moving, well, speed activates everything in the brain. You're just going to see the same hemodynamic response in all of the regions. It's not so interesting. This is completely false, actually. We see subtle differences depending on the different regions that we look at. The first line is the different cortices, and you see that, for instance, the retrosplenial cortex is way more activated than the S1 or the auditory cortex. Now, in the hippocampus, you see very nice activations, and in the other structures below the hippocampus, you see only the dorsal thalamus that's activated during locomotion. So another way to do that is to reconstruct that sequence that we were seeing. And to do that, what we are going to do is compute cross correlations between each pixel in our images and the speed itself. So I'm going to display the function of cross correlation depending on the lag, because as we were seeing before, there's a small delay between the speed itself and the hemodynamic response in the vascular structure. And if you do that, and if you display the cross correlation for all of the different pixels at different lags, you see that you're very able to reconstruct that sequence of activity. And you see that the hippocampus is super strongly activated, and that the dentate gyrus, which is the subfield of the hippocampus, does not activate at the same time as the CA1 and CA3, which was to us super interesting, actually. I still don't know what this happens. So we could do the same with a regional cross-correlogram, and you really see the spread of this activity both in coronal and diagonal planes. The last effect that we saw is that we see actually a time effect. We do not see the same activation between the early runs. If we group our runs in the first 15 minutes, the intermediate runs, the next 15 minutes, and the last 15 minutes of our recordings, we see that the activity in the hippocampus strongly and steadily builds up. And you see the inverse effect in the neocortex, which is also a surprise to us. And it really says basically that you have to have this kind of resolution to look at the level of different runs to be able to see these kind of things. So that's it for the multimodal imaging during locomotion. Now let's wrap it up. So multimodal fUS LFP imaging, the idea is that you can monitor brain hemodynamics in distributed regions together with neural activity in behaving animals. And so far, it's the only technique that can achieve such a large field of view with minimal constraints on the animal. There's 3 different challenges to overcome. So I'm not going to detail on that. During REM sleep, we see brain-wide events that we call vascular surges. They're tightly coupled to the hippocampal rhythms, especially gamma rhythms. And during locomotion, we compare the activity at the level of single trials, and we have a strong modulation of hemodynamics, despite the fact that I didn't show it here, but the behavior is super stereotyped. Even if the animal runs at the same speed, we see that modulation of hemodynamics. So I would like to thank Iconis for inviting me, all of the people that I've worked with through the years. So during my PhD, Ivan Cohen and Lee Manasio were very important to all the different efforts that we put in imaging on mobile animals. Of course, I want to thank Michael for his support and all of the different projects that we've developed through the years, and time passes very fast. Now we realize that. So thanks, Michael, and thanks, Marta, for, during your PhD, all of the amazing work that she did. And again, go to see the publication we have together. We have some super cool results. So yeah, thanks again. I'll pass the floor to Andrew, and I'm ready to answer questions.
Okay, thanks very much, Antoine and Davide, for those fantastic presentations. We'll just give you a brief moment to catch your breath and perhaps a drink before we jump into the Q&A. Just a reminder to our audience, if you have a question you'd like to ask, you can submit it using the Q&A tab on the right of the screen. We may not have time to cover everything today during the live session, but we'll be releasing a Q&A report within a few weeks, so please stay tuned for that. Let's just bring Davide and Antoine back on here. All right. Hi, guys. Welcome back.
Hello.
We've received quite a few questions already, including several from the registration form. So thank you very much to everyone for submitting those. For our audience, if we do run a bit over time today, just a reminder that, and you have a prior engagement, just remember that this webinar will be available on demand sometime in the next 24 hours. And we've also added many of the papers, reviews, links that Davide and Antoine mentioned during the presentations in the handout section. And these will also be available with the on-demand recording. So that said, let's jump right in. And our first question here, just out of curiosity, Davide, you mentioned that the choice of the olfactory bulb was unfair for bold fMRI. Maybe you could just elaborate on that a bit to kick things off.
Yeah, no, it's just because it's both. It's olfactory bulb and mice. So, it's a very, very tiny volume. We are talking about 2 millimeters square for imaging. And so it's really super tricky to make a shim properly, in particular if you are working with a ultra high magnetic field. And moreover, also the PMP or TPX, whatever you want to call it, cover that is suitable for functional ultrasound, although it does not show any artifact for both fMRI, it breaks the homogeneity all over the olfactory bulb, and so it makes the shimming even trickier if it is possible. So if you use, for instance, the same cover in the neocortex, over the neocortex, you can shim fairly well, pretty much the same way you would shim in a naive mouse with no craniotomy, no cover. But the olfactory bulbs were really, really tough, and that's why we couldn't manage to go as fast as we wished with both fMRI.
Okay, great. So basically, it's very technically challenging, ultimately.
Yeah, it's really technically demanding. Yes.
Okay, wonderful. From the poll results, I see we have quite a few electrophysiologists joining us today. And we've had a number of questions related to interaction between the fUS and the electrophysiological signal, Antoine. So perhaps you can expand on that. Do the two interfere with one another in any way? How the fUS recording may affect the ePhys and vice versa?
Yeah, so it's a good question, and actually it's something that we cannot completely rule out. We are sending ultrasonic waves. There are biological processes at stake here, and ultrasound wave actually do modulate brain activity. So far, when the electrophysiology is properly grounded, we don't see any difference visually. And it was also important to do that approach where we would have both the probe and the electrodes on the same plane, and other recordings when we don't have that and we don't see any change in the electrophysiology signal. And the second thing to keep in mind is that we are mostly using planar waves, right? So with focused waves, you can definitely change the pressure, perhaps even trigger electric activity. Here, I don't think that occurs. And when the animal is not grounded, you actually see the ultrasonic bursts. So if things are done properly, I would not worry too much about the two interfering.
Great. Okay, that's fantastic. Question again for you, Davide. In previous papers, for example, on fMRI, many showed that the relationship between stimulus strength and the bold response is highly nonlinear. Instead, you found a linear relationship. Can you comment on that?
Yeah, in fact, that's a good question because there is a quite bad habit, to my point of view, in neuroimaging to consider pretty much the stimulation pattern the same as the neuronal activation pattern. And this can be acceptable in a first approximation if you are dealing with something which, for instance, with a slow temporal resolution. But if you can sample fast, and if you are in a condition in which, for instance, in our case, the neurovascular coupling is fast, as we found, then you can no longer make this assumption. And so in our case, we demonstrated that the nonlinearity was completely made by the neuronal encoding. So if I had to plot the stimulation strength in terms of a boxcar function versus the neuronal responses, I would have found a logarithmic relationship. What is actually linear is the neurovascular coupling that I recall. It's not the relationship between the stimulus and the vascular response. It's the relationship between the neuronal and the vascular response. So that is the tricky point. So we should really pay attention to that. So the good news are the neurovascular coupling is linear, at least in our condition, in many other conditions that we could test it in our paper. But please don't take just the boxcar to rule out information about the linearity of the brain.
Okay, fantastic. Another question for you here, Antoine. Great talks, agreed. Would you care to speculate about the relationship between hemodynamics and gamma? You mentioned that gamma could be causal, but how might one test that? And lastly, theta also couples with hemodynamics, but is less dynamic.
It's a good question. So I might have gone a bit too far saying that it was causal because I haven't proven it. We see correlations. We also see a delay, and we see that the gamma activity precedes the vascular events. It's not only me who's related gamma activity to blood flow. There's been several papers about it. When you try to explain, it was mostly done with bold fMRI. So when you try to explain the variance that you see in bold fMRI studies, the best regressor is mainly the gamma activity. Now it's different bands depending on the species. It's not exactly the same processes. But to me, so far from my understanding, I would go towards the role of inhibition and interneurons in the brain because they are super involved in these gamma cycles and these gamma oscillations. And most of these neurons are known to influence neurovascular coupling. So the next step, I guess, is to try to investigate this causal relationship and maybe block gamma and see the effect on the vascular surges.
Okay, fantastic. Maybe either of you or both can answer this question. We've had a number of questions related to the resolution, spatial and temporal resolution that's achievable. And this one specifically asks, is it possible to obtain resolution at the level of blood vessels in the choroid plexus?
Wow.
I would
That's
say
a tough
yes,
one.
but I'm not an expert. I see where this comes from, and I think it's a super important subject. The link with cerebrospinal fluid and what we see is obviously an important question. If the vessels are not too small and they are the size of roughly what you can find in the normal vascular and brain structures, I don't see why we would not be able to see them. We can also investigate movement of bigger structures and especially the movement of choroid plexus, which is something also currently under investigation. So I don't see any reason not to see them if they're not too small.
And if I can complete, there is also this new, high-resolution functional ultrasound that has been just introduced last year by the group of Michael Tanter. So probably with that approach, we will get chances to look at what you want to look at.
Okay, excellent. We are just near the hour mark here. As I said, we'll try and get through as many more of these questions as possible. If you do have to leave us, thank you very much. The recording will be published on demand shortly, and I've launched the survey. If you could take a moment to fill that out as well, we'd greatly appreciate it. We do read all of your answers. Another question here for Antoine. Is it possible to use standard silicon probes, such as Cambridge Neurotech, Buzaki probes, NeuroPixels, in combination with functional ultrasound?
Yeah, quick answer, yes, definitely.
Okay, fantastic. And maybe along those lines as well, what is the biggest obstacle to safeguard the implant's integrity for extended periods of time? So maybe weeks, months.
That's also an important question, but it's trickier and it's something that everybody in behavioral neuroscience would struggle with. We're actually discussing that with Davide just before the webinar started. So I would say one important stuff is, of course, handling and having the animals not being too stressed and removing whatever that's in the cage that could cause trouble to the safety of the implant. Now, it's really also about the design. If you have anything that's too bulky, you increase the chances that the animal would actually scratch their head or just bump into things and exert the mechanical stress on the implant. So I think the design is important. The fact that the animals are well and do not feel that they want to scratch themselves too much will definitely increase, and you'll be able to keep your animals for way longer than if they're stressed. So it's something that's definitely a bit overlooked in neuroscience because it requires time, but it's definitely worth investing the time to do this.
Okay, fantastic. Another question here. We've had a number of questions about the craniotomy. Does the craniotomy affect the intrinsic brain activities? Is it possible some unexpected changes in brain activity could be attributed to the craniotomy and associated inflammation, I guess? And I'll also expand that to we've had some questions about whether or not it's possible to do this through the skull with an intact skull. So perhaps either or both of you could elaborate on that as well.
Do you want to take it, Davide, or?
I can start at least. So,
sure.
Of course, no argue. The craniotomy is an invasive approach. So for sure, we are not dealing with a brain which is completely preserved. But on another point of view, it's also, it's the two things. We do the craniotomy and we use these covers in order to run experiments over time. So we give the mouse or the rat enough time to recover not only from the surgery but also from any kind of manipulation that we can do of the brain. And another advantage that can be feasible with this cover is that they're easy to bend, and so we can really follow the curvature of the brain without changing the shape. Then, of course, you can deal with troubles in temperature. We also made a paper on the temperature, not with functional ultrasound, with 2-photon microscopy in Social Park Lab. It's Morgan Roche et al. In eLife 2020, I think 2019, something like that. But still, apparently the brain showed to be quite resilient to these changes if you are looking at the brain after enough time in order to give it time to recover. And through the skull instead, I think you have to refer to the papers by Sophie Pézet, who did a very nice imaging through the skull. If I correctly remember, it's completely feasible in mice. In rats, it's okay for the neocortex. It starts to be a little bit less performant with respect to a craniotomy in the thalamus. So if you are willing to go deep, and then it's another question about the reliability over the time. This is something that with a craniotomy you can just look through and see how the brain looks like. Without a craniotomy, you are not sure. I mean, I wouldn't be sure about, but I'm talking about very large time intervals, so months from mice young to adults. This
Fantastic.
is my experience.
Did you want to add anything to that, Antoine?
There was a very complete answer. Just to mention that, of course, the skull of rats here is very thick compared to the one of mice. So you might be tempted by only thinning the skull, but then there's a trade-off in the temporal window that you'll get good signals. So I'm a big defender of this craniotomy approach because we invested a lot in improving them. And so far, when we also try to check for inflammation, we don't see obvious markers of inflammation for extended periods of time. So sure, there's also the problem of temperature that Davide mentioned, which is definitely something that could modulate brain activity. We are not the only ones doing this kind of craniotomy. Maybe we do definitely bigger ones, but we are not the only ones to do this, and it's been investigated elsewhere. So I would not be too worried, but it's important to keep in mind. You also have the visual inspection, and sometimes you see that the tissue is basically, you want the brain is starting regrowth of the different tissue, and you have something akin to skull that's developing beneath the implant. So yeah, that's more or less it sums up the issues with craniotomy, I think.
Okay, that's fantastic. Great answers. There's one question here about connectivity changes when the animals are anesthetized. I guess typically the more data has historically been available on the anesthetized animal than in the awake. So perhaps this question is related to the validity of data collected from animals who are anesthetized. But perhaps you can comment on that as well.
Well, anesthesia is a very strong modulator in brain activity, and it's most of the time it masks many. For instance, when we do sleep studies, it's two complete different states. So we are actually looking for a technique where we can not use anesthesia, and we can actually compare in the few of the recordings that I used to compare. We definitely do not have the same correlations, do not have the same baseline, and we see the time that we need to wait to have a signal going to baseline either after the animal woke up from anesthesia.
Okay, fantastic. Maybe we'll make this the last question. We've gone quite a bit over for time here, and I appreciate the audience and your attention and participation today. Is it possible to combine the fUS with miniscope or optogenetic stimulation approaches? Perhaps you can add something to that.
Yes. And optogenetics, it's been done already. There's a paper published in Neuron 2021. Arnaud Sandublanc is the first author of that study. And with miniscope, nothing prevents. Miniscope can be a bit bulky, so it would be 2 probes, but on the 2 different sides of the brain in rats, I think it's definitely feasible, yes.
Okay. Fantastic. I just, unfortunately, we're out of time. I think we could probably go on for much longer. But I'd really like to thank you, Davide, Antoine, for the fantastic presentations, your time, and also sharing your insights with us today. It's been an absolute pleasure having you with us.
Thank you very much,
for the invitation.
Thank you very much, both. For everyone in the audience, a big thank you as well. I really appreciate your participation today. And last but not least, of course, we'd like to thank the sponsor of today's event, Iconius. without whom this would not be possible. And ultimately, in closing, we hope you enjoyed this Inside Scientific webinar, and we'll see you again next time. Thank you, everyone.